Hi everyone, my name is Takunda and today I'm going to talk about my presentation on NeuroAI from animal replacement to computational psychiatry partner as the title. This presentation focuses on evaluating the role of generative models in future of psychiatry research. So I'll start by expanding more on the bedrock of research, thinking about animal models in psychiatry. So for decades, animal models have been the experimental backbone for studying psychiatric disorders, as they allowed for the controlled manipulations to study the neurobiology of stress, behavior, and neurochemistry. And some key examples I have here are the use of rodents and avian species to study the HPA axis activity. So the HPA stands for hypothalamic pituitary adrenal axis, and this is responsible for the cortisol or corticosterone regulation. Corticosterone is a primary biomarker for allostatic load and adaptive capacity in avian stress physiology. And we also see paradigms like chronic unpredictable stress, lent helplessness, and novel suppressed feeding assays within the rodent species. So I'll dive into the crisis of translation and the call for alternatives, which is the link between the use of animal models and in situ models. So notoriously, there's been a high translational failure rate when it comes to using animal models and thinking about how we can translate that to research in humans. There have been some scientific limits, including reproducibility crisis and significant species differences when it comes to neuroendocrine systems, and this limits the translational accuracy of the work. In addition, I would want to talk about some conceptual gaps. There have been some difficulties in modeling complex human psychiatric constructs, for example, like rumination or social withdrawal. These have been kind of like tough to work with when you are thinking about animal models, as these two systems kind of operate differently in some ways. There's also some ethical imperatives we have to discuss about. We have some growing concerns and the three R's principle, which is defined as the replacement, reduction, and refinement aspects of research. And these have been spurring the search for computational alternatives. So that's where the challenger emerges, and we have generative neuro AI, which has been on the rise in this past couple years. There's been a paradigm shift from observing biological systems to simulating them. And generative neuro AI aims to build synthetic agents that mimic complex behavioral and physiological trajectories. Three examples that come into play I would want to just like mention as we're talking about this new approach. We have some agent-based reinforcement learning that works on data provided for these models, and that information is kind of like used to reinforce the learning aspects of some computational models. We have the digital twin models, which has been researched on and written like several papers, and we also have some generative agents that are mainly used to generate data that can be later used by some reinforcement models. So I'll talk more using an example of modeling stress and behavior trajectories in silico. So Neuro AI shows promise in generating synthetic data that mirrors biological processes, particularly in stress physiology and decision making. An example is simulating dopaminergic reward prediction error signals. We can also think about building digital organisms with parameters that are analogous to HPA axis reactivity. Through these activities, you'll be able to generate like some response curves that kind of like resemble corticosterone modulation. So in this figure I have here on the x-axis, we have the time post-stressor, and we have the corticosterone levels on the y-axis. The blue line represents the actual biological data, and the gold broken line represents the NeuroAI generative model simulation. So you see these two lines kind of like fit on top of each other, the biological one and the simulated one, meaning that this data can kind of like be related in some way when it comes to fitting the data, and there is some simulation similarities you can see between the two different stress responses. That takes us into the next question. The critical question we have here is, can a simulation capture biological aspects that might be happening behind the scenes? And are these models approximating the phenomenology or the biology? While current AI excels at fitting data distributions and generating plausible behavioral trajectories, the key challenge is emulating true mechanistic adaptation. That leaves us with a question: can a synthetic agent learn genuine physiological resilience or dysregulation over time, or it is just replaying patterns from its training data? This is a very interesting question. Let's do a reality check. When we are thinking about generative models, they primarily excel in behavioral level abstraction. They generate trajectories consistent with observed data. However, they do not capture the underlying biological machinery unless explicitly programmed. As you can see in the figure, we are able to capture the visible or the simulated aspects, for example, behavior. We can also think about decision making, and we can also relate to the example I've provided of the simulated stress response. However, we still have some hidden or missing layers we need to understand and think about. For example, we don't know what's happening within the genomic regulation. We don't know anything about the mitochondrial stress buffering. In addition, there is no information to help us understand the cell level receptor sensitivity, neither the receptor kinetics or immune endocrine crosstalk. We have new tools, but we also have new risks. So let's expand the ethics in silico psychiatry. It is important to understand the epistemic risk of using generative models. They could reinforce a black box approach where surface level prediction replaces causal mechanistic understanding. Also, there are some algorithmic biases that can come with these generative models. The risk of replacing natural biological variability with overly deterministic, clean, and potentially biased computational representations shouldn't be overlooked. There is also, in addition, some oversight gap. Ethical oversights for AI modeling is really underdeveloped at the moment. There are no equivalent to institutional review boards that exist to govern potential epistemic harm of mismodeling psychiatric disorders. This is a critical gap that needs to be addressed. We get to the crossroads where we have some imperfect tools, but also need to make strategic choices. Animal models suffer from translational failure and rely on arbitrary stressors that may not map to human conditions. On the other hand, we also have NeuroAI models which lack biological embodiment and introduce new epistemic risks. The crossroads we are at here encompasses the non-biological limits and the unknown algorithmic risks. So the goal is not to ask if AI can perfectly simulate biology. The question we have to ask is, can AI surpass our current biological approximations in translatability and ethical optimization? I would want to talk about synthesis, thinking about a hybrid neuro AI biological pipeline we can use to synthesize all these aspects I've been talking about and understanding how we can make the best of this situation. The most productive path forward is not replacement, but an iterative feedback loop between ethically constrained biological validation and advanced computational modeling. Because we have to use computational approaches to advance our science and make use of the new technologies that are emerging in today's world. So I have a four-step approach where we can use some small-scale biological sampling and we get some data that can be fed into generative models. After that, we can use AI to interpolate and expand trajectories to give us some insights that we can use to predict and guide next experiments. Through this, we will move from thinking about replacement into understanding computational amplification of limited biological data. Through this hybrid pipeline, we have a four-step iterative process. The first step will be seeding, where we conduct a small-scale ethically monitored biological sampling at high quality using mechanistically relevant data. And after that, we will train our generative models. We will use this sparse but rich data to train and ground generative models, embedding real physiological parameters into reinforcement learning agents. Once we are done with that, we can amplify the data. We will leverage the trained AI to simulate and interpolate and expand these trajectories across vast digital populations, exploring parameter spaces impossible to test in vivo. Once we are satisfied with that, we will be able to refine. We will use insights and predictions from the in silico models to design the next, more targeted and efficient minimal biological experiments. So, in a hybrid pipeline, the goal shifts from routine large-scale animal experimentation to sparse, strategic, and high-impact biological validation. We will be able to use less model organisms, thinking about the 3 Rs I used earlier. However, we'll still be able to benefit and make use of the computational approaches we have to kind of like help with the reduction aspects of the, for example, how much animal models being used within this different kinds of research being conducted. So animal models will no longer be the primary discovery engine. Instead, they become seed generators for AI-driven predictive ecosystems. The future is exciting. The future of discovery is going to be talking about NeuroAI as an epistemic partner. Generative NeuroAI models are a paradigm shift, but their current limitations prevent them from being a direct replacement for animal research. We can't replace animal research fully at all, but by integrating them into a hybrid pipeline, we will be able to leverage the strength of both worlds, the biological grounding of in vivo systems, and the scalable predictive power of in silico models. In this future, NeuroAI is not merely a tool of simulation, it is an epistemic partner in the complex quest to unravel the mechanisms of stress, resilience, and psychiatric adaptation.